Okay, welcome everybody. Good afternoon. Welcome to the Medical Scholars Series, jointly sponsored by the Fralin Biomedical Research Institute and the Virginia Tech Cardiology School of Medicine. By way of reminder, this speaker series honors Dr. Timothy Johnson, one of the founding faculty members and leaders of the medical school, who was very much committed to the nature of this type of program and the research component, and having key intellectual thought leaders from various places come in and speak to the medical students and the graduate students about their work. So we like to honor Timothy Johnson every time we have this program and remember him. He was certainly an important leader in the formulation of this entire medical school curriculum. So for today's speaker, Dr. Arany from University of Pennsylvania, I'm going to turn it over to one of you, Mason Wheeler, who's going to come to the podium and do the introduction of the speaker. Mason, welcome.
Hi guys, as Dr. Fralin mentioned, I'm Mason Wheeler. I'm a second year TVMH student, and I have the distinct honor of introducing Dr. Zoltan Arany today. Dr. Arany is a physician scientist from the University of Pennsylvania in the Perelman School of Medicine, and he is a leader in the field of cardiovascular metabolism. He received each his BS, his MD, and his Ph.D. from Harvard, and is board certified in internal medicine and cardiology. He's incredibly involved both in his institution, UPenn, and in the scientific field at large. He is the Samuel Bellet Professor of Cardiology, directs the Cardiovascular Metabolism Program, the co-director of the cardiovascular divisions, both of the Institute for Diabetes, Obesity, and Metabolism and Institute for Regenerative Medicine. He chairs the Cell Biology, Physiology, and Metabolism graduate program and is the scientific director of the Penn Metabolomics Core. He's also an ad hoc reviewer for many high-impact journals and consults for many health sciences companies, including Pfizer. As of this week, he's been cited over 20,000 times and has over 170 publications in extremely high-impact journals such as Nature, Science, Cell Metabolism, and Circulation. So without further ado, Dr. Zoltan Arany.
Thank you. Thank you for that intro. Is this working? Yes, it's working. Well, thank you for inviting me. It's really a pleasure to be here. It's a pleasure to be in person. Just love it. So tired of the hybrid scene. I want to talk a little bit about human metabolism. I'll try to be quick. I might have a few slides at the end so we have time for questions. But I also like interruptions. So if you feel like it, just interrupt. I don't mind. It's sort of more interactive that way. But again, thanks for the invitation. And let's dive in. That's a picture that used to be what I saw outside my window, but now there's a big, huge building in the way because as with you, we're in a growth phase, and that's exciting. I think all the biomedical enterprise is in growth phase for a lot of places in the country. This is a really exciting time to go into biomedical research. I'll make that pitch right off the top. We could talk more about that if you want. Okay, this is I've been accused of being unfocused, and it's kind of true. I'm a little bit all over the place, but our lab, the common theme is metabolism. We love metabolism, especially as it relates to the cardiovascular system. I am a cardiologist after all, and blood vessels in particular, although I won't talk about blood vessels today. But today we're going to focus in on heart failure and cardiac metabolism. I don't need to tell you that heart failure is a problem. It's still a problem. We've gotten a lot better at treating heart failure, but it's still a problem. If you walk to the hospital with a diagnosis of heart failure, you have a 50% chance of not surviving 5 years. That's a pretty nasty number. And now we have the new kid on the block, which is heart failure with preserved ejection fraction, which at this stage of the game has the same prognosis. And I'll argue we have therapies for HFrEF. We maybe have one now for HFpEF. I will argue that none of those, so those therapies are obviously not good enough because we still have a 5-year mortality, that's 50%. And I'll argue that none of the therapies we use are actually targeted at the heart. Everything is targeted outside the heart. It's all blood pressure regulation, volume control, et cetera, right? So I think there's still a missing ingredient here. And perhaps metabolism is one of those things. For sure, the heart is wildly metabolic. I don't need to convince you of that fact either. It's the most oxidatively active in the body on a per weight basis. In fact, because of that, it's chock full of mitochondria. So I don't know if you realize this, but if you were to hold a heart in your hand, which you will as medical students at some point, you're actually holding a third of that is mitochondria. It's just pure mitochondria, right? The other 2/3 is, myofibrillar, and so there's a tremendous amount of metabolic activity going on in the heart, and it has a range, right? So it can actually increase quite dramatically during exercise, right? So it's highly metabolic. Is heart failure accompanied by a metabolic problem? There's a lot of evidence to support that that's true. And I'm not going to go over the literature because there's a lot of it, right? I'm just going to give you one example. This is a great, if you want to read a quick review on this question, this is a really good one. The Failing Heart, an Engine Out of Fuel by Stephen Neubauer, which was about 10, 15 years ago. Yeah, here it is, 2000. So there's a lot of different kinds of evidence to suggest that the failing heart has bioenergetic defects. The best kind of evidence is actually this kind, I think. It's a non-invasive NMR and MRI, where you can get spectra for ATP and various different, ATP, ADP, AMP, et cetera, from a patient that's in the magnet without having to perturb them in any way. And you can just look at peaks for ATP and phosphocreatine, for example, and you can do math and conclude what the sort of energy charge is in people as they're sitting there in the magnet. The energy charge in people with heart failure is distinctly lower. So the heart is having a hard time keeping its energy charge up, okay? And that's been demonstrated by dozens and dozens of studies at this time. So it's very clearly true. And not only that, but how the worse that energy charge is, the worse your outcomes is. It's actually one of the best predictors of outcome. It's just not a cheap test, but it's one of the best predictors of outcome in heart failure. This is why we're interested in cardiac metabolism, right? We think it's a player in heart failure. So I'm going to tell you 3 quick stories about this. The first 2 are going to be in humans. One's going to be in mice, partly in mice. Let's talk about fuel use in the heart. And then I'll talk a little bit about looking inside the failing heart. And then I'll just take one example, branched chain amino acids, to tell you how we want to then take this information and learn more about cardiac metabolism. And all 3 of these stories, these last 2 stories actually just got published. I used to say they're unpublished, but they just got published. This is also exciting. And I'll tell you the references. Okay, so metabolically speaking, the heart is often called an omnivore, meaning that it likes to eat anything it wants, which is kind of true, but not totally true, as I'll show you in a minute. But there's a gazillion, don't squint at the details. There's a lot of diagrams that look like this in literature. I always pick on my friend Gary Lopashuk, but I could really pick on other people too. The heart likes fat most, and then it also eats some glucose and some amino acids and some ketones and some lactate. And for the most part, it all goes into the TCA cycle to get burned because that's the efficient way of generating ATP, right? Rather than glycolysis. In heart failure, there we go. In heart failure, this changes. Fatty acid oxidation is not as efficient. Glucose oxidation goes up. Lactate goes down. Ketone oxidation goes up. At least that's the party line, okay? This is what most of the diagrams are going to look at. But I'll submit to you that this is based on, hundreds of studies, 95% of which are in rodents, sometimes rabbits, sometimes dogs, okay? How do we know that this is actually true in humans? Because it's all based on model organisms. And that was a question that was of interest to us a few years ago, and we wanted to tackle it. So how do we know? So we're not, it's not like we're the first people to ever look at humans. They have been studied. And I think the best data so far on how we know this to be in humans is using coronary sinus catheters, okay? So catheterization, meaning putting a tube, putting a catheter percutaneously through the groin, let's say, for example, and threading it into the heart, is not a new invention. It was invented in the 50s by this guy, Theodore Forsman. Now, this is a funny little side story. When he came up with this idea of putting a catheter up the vein and all the way into the heart, people thought he was completely crazy. No one would let him do it. In fact, to the extent that eventually he decided to do it on himself to prove that it could be done. right? So this is the first picture of the first X-ray of a catheter in anybody's heart, and it is his heart, and he put it in himself and then ran to the X-ray lab to take. You cannot do this anymore. This is a 50s thing, right? Right? Nobody can do this anymore. Anyway, now we're more sophisticated. Of course, we put them exactly in the coronary sinus. So why the coronary sinus? Because the coronary sinus is great. The coronary sinus drains just the heart. It drains the heart and nothing but the heart. Right. So that means that if you are able to measure what's going on in the artery and you're able to measure what's going on in the coronary sinus, then the difference between the two is something the heart did. If there's more in the sinus, that means the heart spit it out. If there's less in the sinus, that means the heart used it. Okay. So you can use this to quantify flux of stuff, right, in and out of the heart. And we're not the first to think of this. There's been about 50 studies in the 70s and 80s. They're usually using rudimentary kits. Nobody had mass spectrometry back then. They're very low numbers. They're usually done in 18-year-old healthy male volunteers because that's what you did back then. So we decided to revisit this with a modern era, and that's the first story I'll tell you. So we teamed up. So this is all the work of Danielle Morshiga, who's just an amazing MD Ph.D. student. She's now back in the clinical wards. We teamed up electrophysiologists named here. Patients are coming to the EP suite for elective procedures, atrial fibrillary ablation. So we're not, these aren't volunteers. We're just piggybacking on clinical care here. And they all have catheters placed in their coronary sinus as part of clinical care, as well as an arterial line and a venous line, a femoral venous line. So we're drawing blood simultaneously from here and the coronary sinus and the artery. And we did this in 120 patients, 80 of whom had normal ejection fraction. And then we also picked 40 that had low ejection fraction because we thought it'd be an interesting comparison. And it was, as you'll see in a minute. And then we ran those samples through mass spectrometry, looking at 300, 400, 500 metabolites at a time. We did a lot of work with Chelsoon Jang, who was an awesome student in my lab, and then he postdoced at Princeton. Now he has his own lab at UC Irvine, and we still work with him. He's quite brilliant, actually. Fly through this data because it's published a couple of years ago now, or a year and a half ago. But just to show you, just to give you a flavor, right? So that whenever you see a clinical study, always look for table 1, because table 1 should tell you something about the patients that are getting studied, right? Because you want to know who you're studying. Okay, so again, don't squint. The point here is that these were basically the average American in his or her 60s. Actually, it's amazing the extent to which these demographic match the average population in the United States. So what does that mean? So as I said, in the 60s, if you're in your 60s in the United States, your average BMI is 30. It turns out. It turns out that a fair number of you of us have hypertension. A fair number of us have diabetes. Our fasting glucose is kind of up a little bit. Our home IR, which really should be 1, 1.5, is up a little bit as well, right? So this is the average person with comorbidities, okay, and a relatively high BMI. So very different from that, 18-year-old volunteer in the 1950s. Very different person. And then we run, we do the mass spectrometry, and we get tons and tons of amazing data. And we do all these volcano plots that look like this. Again, don't squint. I just want to get the point across. So I'll go through some examples in a minute. But volcano plots, if you've never seen them, they are typically, this is fold change on the x-axis. So stuff on the right is released by the heart, stuff on the left is taken up, and then the y-axis is your confidence, is the negative log of the p-value. So some of this stuff has got p-values of 10 to the minus 10, 10 to the minus 12. It's a very robust data set, it turns out. We're very sure about a lot of stuff that's in here, okay? So what are some of the things that are in here? Okay, so one of the first things we can do is we can carbon count, all right? Because we're thinking about fuel and we're thinking about stuff going into metabolism, right? So it doesn't make sense to molecule count, because one molecule of fat is going to be metabolically very different than a molecule of sugar, which has only 6 carbons versus 16 carbons, right? So we carbon count as a kind of a way to, match everything, right? It's not perfect. I'll come back to it in a minute, but we can carbon count. And, above this line is taken up, and below this line is released. So the heart takes up a lot of carbon. That makes sense. Turns out that most of the carbon it takes up is fat. And then it likes to take up some ketones and some lactate and some amino acids. And then it also spits out some amino acids, right? And so we learned a lot of interesting things from this. For example, it loves to spit out glutamine, a lot of glutamine, and that has led to some interesting studies about how glutamine being spit out by cardiomyocytes talks to fibroblasts. I'm not going to talk about that today, but we worked with John Elrod at Temple to do some of those studies. That's really kind of fun. So that's carbon counting, and I'll come back in a minute to interpreting this data. We can also nitrogen count. Why not, right? If we nitrogen count, it turns out that the heart is in a major nitrogen deficit not deficit, but negative net negative nitrogen balance is the word I'm looking for. In other words, it's spitting out a lot more nitrogen than it's taken up, which is, I was shocked by this because the carbons I understand, because they're taking up the carbons and you burn them, right? You burn them in the TCA, give them off as carbon dioxide. But where's the nitrogen going and coming from, right? All this nitrogen is coming out of the heart. And there's really only one explanation, which is the heart is being catabolic. It's basically breaking down its protein, which is the only source of nitrogen in the heart. So under these conditions, in these patients who are coming fasted in the morning, right, they are breaking down their protein a lot, about 10 times as much as the leg at the same moment in time, right? And you know, if we let so during those 12 hours, if you do the math, they basically broke down about a couple of grams of their heart. That's a lot. That's a big chunk of the heart, right? So I was very surprised by that. Now, obviously, they must be remaking the protein during the daytime when they're fed, because you know you can't maintain this permanently. But I had no idea about how drastic the cycles of breaking down protein and secreting protein in the heart. So that was an interesting observation. Okay, so then you know you can read the paper. There's a lot of details. I'm just gonna sort of give you a few of the punchlines. So you can do a lot of math, and then you can come up with a pie chart that looks like this, that basically says, okay, these hearts in these conditions are generating ATP, and they're generating ATP from these nutrients after doing much math. So again, most of it is fat. There's an unexplained component. We know it's unexplained because we measured oxygen in the artery and in the coronary sinus, right? So we know what the oxygen difference is. So we know what the carbon difference should be. And we didn't account for it all. And I think this is probably all lipoprotein particles, because that's one thing we didn't measure, right? VLDL, LDL, we didn't measure that. There's fat in there and the heart can't take it out. So I think that's probably what that is. And then, as I said, you've got some ketones and some amino acids and some lactate. Now, I told you we had oh, and the other thing that's interesting, am I advancing here? Right, so lots of fat, probably some lipoproteins. It's a net consumer of lactate. I'll come back to this. A lot of times people think that we talk about glycolysis and secretion of lactate. The heart does not secrete lactate, not net secretion. It consumes lactate. And then it loves ketones, active proteolysis we talked about, and then no sugar. There's no sugar on this pie graph. Right. This I constantly get 1,000 questions about this at these, you're laughing, right? Because it always comes up. I'll come back to that in a minute, but no sugar. Okay, I told you we had 40 patients that had low ejection fraction. Did they look any different? Now, these are compensated, right? They're not in florid heart failure. They're coming to the cath lab. They're compensated, but they have low ejection fraction, right? Do their numbers look different? They do. It's interesting. They definitely slow down their fatty acid uptake a bit. And they pretty significantly increase their ketones, their lactate, and their amino acids. So they're switching their fuel preference. It's definitely true, and that was all statistically significant. So less fat, more lactate, more ketones, and more amino acids. So this was the picture I showed you before, right? That was from rodents. I don't think that's all quite correct. I think a lot of these arrows, or at least, are not showing us the same thing in these humans. Right? We're not seeing a reduction in lactate oxidation. We're seeing an increase in lactate oxidation. We're seeing no glucose at all, for that matter, right? And we're seeing no glucose oxidation. So this is just one study, right? It's one study. Average Americans in their 60s with compensated heart failure who are fasting. Very different from a otherwise healthy rat who may or may not have been fasting, who probably didn't have a lot of comorbidities. So there's caveats to this. But it's a very different study, and it tells us something very different. And I think we need to take this, go back to the rats and to the mice, and adjust our experiments to be more representative, I think, of what's going on in real life in humans. So that was that study. And again, I skipped a lot of details for the sake of time. But the next question was, this is all looking at artery and coronary sinus. This is all inferring what's going on. Well, it's calculating flux, but we don't actually really know what's going on inside the heart. Right? What is changing these fluxes? Why is the heart changing these fluxes mechanistically? And we can't know that from these data. This is just look at before and after. So we decided that we had to look inside the heart. Right? That's the only way to do this. So to do that, we teamed up with Ken Margulies, who's just 2 doors down from me. Ken is a heart failure doc, transplant doc, who amazingly, for the last 20 years or so, has been collecting hearts after transplant. So heart gets transplanted for some sort of non-ischemic cardiomyopathy or something like that, and he's there with the bucket and catches the heart. And he literally has, I kid you not, thousands of hearts frozen away from over the years. Ken Beatty is his partner in crime. So we went in there and looked at those hearts and said, let's look at the metabolism of these hearts and see what we can learn. Right? And this study was just published literally a few weeks ago. So we've got a whole bunch of hearts. What's the number? It's up there, right? So we got about 40 hearts that are post-transplant, non-ischemic dilated cardiomyopathy. And then we compare them to gift. So we got to have a control, right? The control is in essence discarded donor hearts. So somebody has a motorcycle accident, unfortunately, and is brain dead, and the family or the subject donates their organs, but the heart is not the right size, or for some reason it's not used, and so it's to be discarded. And again, Ken is there with both Kens are there with their buckets. So a couple of, and then we did metabolomics and RNA-seq on them to compare the two. A couple of important points about this. It's when you look at these studies, the heart is very metabolically active. If you take the heart out and you put it on the table and you wait for a minute, everything's changed. If you wait for 10 seconds, everything's changed, right? Ischemia changes metabolism in the heart almost instantaneously. So it's extremely important how they harvest this, and you should always look at the methods in papers that talk about this kind of stuff. What the Kens do is they put the hearts under cardioplegia before they ever extract them. Right, so the heart is basically never under warm ischemia, right? And that's true of both the donors and the transplants. So these hearts are very, very carefully harvested. So that's important. And the other thing that's important is that the sample is always from the same spot. It's not like right heart versus left heart versus septum versus apex. It's always the same spot. So these things matter actually when you do this kind of study. Okay. And then this is all non-ischemic dilated cardiomyopathy. So we're not talking about ischemia. That's for another day. Okay, you get volcano plots again. Same thing. Again, 10 to the minus crazy p-values. It's really beautiful data. Also here we have plasma, which is kind of cool. We didn't have, we can compare the plasma to the tissue. If the plasma and the tissue say the opposite thing, that tells you a lot. It tells you the tissue is doing something very specific. So we have lots of things. So let me run you through some of this data. And I'm gonna, so first sanity check. We have a metabolic defect in these failing hearts. We should, right? If all prior literature is right, and so that's what we see. We see ATP is low. We see phosphocreatine, which is an energy charge, is low. The ratio of ATP to ADP is low. The ratio of phosphocreatine to ATP is low. So sanity check, right? Our failing hearts are showing a bioenergetic defect. So that fits with previous data. So now the way I'm going to run through some of this data is by taking the previous study, the observations in the previous study, and looking at what can we learn, how does this study inform the observations from the previous study, right? So I told you there's less fat in the previous study in the heart failing versus the non-failing, right? So why is that? The party line is that there's a fatty acid oxidation defect. There's a problem with fatty acid oxidation and mitochondrial function in heart failure. That's what all the reviews will tell you, right? I'm not sure that that's what we're seeing, honestly, because here's a diagram of fatty acid oxidation, right, going on in the mitochondria. So fat comes in, and then it's a CoA moiety is put onto it. So now you have acyl CoAs, and then the acyl CoAs are transported into the mitochondria through a transporter that uses acyl carnitine. So you go acyl CoA to acyl carnitine back to acyl CoA. And then that acyl CoA inside the mitochondria gets all fatty acid oxidized, 2 carbons at a time, right? You just got through your biochemistry lecture recently, I bet. So if you had a problem over here, you would expect accumulation of all the stuff upstream. You should see more acyl carnitines, you should see more acyl CoAs, you should see more fats, right? We see exactly the opposite. Everything is way down in the heart, everything. All the fats are down. All the carnitines are down. I'm not showing you, but all the CoA species are down. Everything is down, right? So to my mind, that doesn't make sense. To my mind, the problem is up here. And we know very little about what's going on up there. So this is a direction that we're going to take the lab. Now we're going to have to go back to the drawing board and try to understand it. Yeah.
Oh, did you see downregulation of CD36?
Not at the gene expression level. Now we have to look at it better. We have to look, so subcellular localization, we haven't looked at yet. Post-translational modifications, we haven't looked at yet. So this is the direction we want to go. But at the gene expression, it looks fine. Yeah. The only thing we saw that was down was ACL1, which is up there, but it still doesn't explain why the fats themselves are down. This is downstream of the fats. So this is a question that we now want to take to the next step. I don't know the answer. How about this question? The lactate, why did the lactate use go up? For this, I think I do have an answer. It's actually interesting, also kind of anti-dogmatic. But I think that, so here's glycolysis. I'm sure you recognize it more or less, right? And lactate is secreted in many cells like cancers, but here it comes in, right? And it comes in and then it goes into the mitochondria through the transporter. And the rate limiting step of pyruvate use is this PDH complex, pyruvate dehydrogenase complex. And I think that spigot is open in these failing hearts. Why do I think that spigot is open? Because the inhibitor of PDH is this molecule called PDK. It's a kinase, inhibitory phosphorylation site. And that thing is gone. It's gone at the RNA level. It's gone at the protein level, right? We've done the Western blast, et cetera. It's gone. And if it's gone, you would expect this complex to not be phosphorylated. And that's exactly what we see. The phosphorylation is down, and you'd expect the activity to be up. And that's exactly what we see. The activity is up. So I think this spigot is open in failing hearts. Now, is that a good thing or a bad thing? Is the heart, adapting or maladapting by doing this? We don't know the answer. We're not sure about that. So this is something we have to go back and test. Although I would argue the data so far would argue that this is an adaptive process. But I think the spigot is probably as open as it can get. So I'm not so sure that we can give drugs to open it even more, which has been tried actually and has failed. So I think I have an answer to that one. Why more ketone use? That's a big change, 6 to 16. And ketones are hot these days. There's a lot of hypotheses about ketones being good for failing hearts. So this might be an adaptive thing. So how does the heart do this? Go from 6 to 16%. I think our data supports the idea that the heart doesn't do this, actually. It's not the heart that's doing this, it's the body that's doing this. Because the plasma ketones are markedly elevated, just as much as the tissue ketones are elevated in these patients. And if you look at all the patients, we had lots of patients, right? If you look at all these, going back to the AACS study, if you look at the 120 patients that we studied there, and you ask, what is the correlation between the plasma level of ketone and the amount of ketone that was taken up by the heart? And that correlation is perfect. It is nearly perfect. In other words, I think that the heart basically takes up exactly the amount of ketones that it's faced with. So there's no regulation step, right? So the reason that the failing heart is taking up more ketones is just because there's more ketones around in a failing heart person, right? So it's not the heart deciding anything. It's probably the liver, probably, but to be proven. But the liver is thought to be the major site of ketones. Yeah, which is an interesting question because it leads to the idea that a failing heart has an impact on liver function, which is an understudied domain that we're also interested in pursuing. Yeah. That's a good question. So completely different from opening the spigot. Here, there's no spigot. It's basically just taking what you get. Right. So it's very interesting to think about the different mechanisms of, and there's only actually in all those metabolites that we measured, there were only 3 that did this. There's only 3 that were just taken up as they come, and they were the ketones. These are both ketones and glutamate. Right. We can talk more about glutamate if you're interested, but for some reason it just takes up glutamate. So it's an omnivore, sure. Does it actually eat what it sees, which is kind of a party line you sometimes get? Sure, it eats what it sees, but it's not the only entity that decides that. There's ample regulation here, the regulation that we can usurp, hopefully, for benefit. Why more amino acid use? Well, there's more proteolysis, and I'm not showing you the data, but there's more proteolysis, which is interesting, potentially pathologic. And then why no glucose? I said I would come back to this, right? Why no glucose? And I don't really know the answer, but they're fasted. So there's low insulin state. So that could be one reason. They're also all hyperlipidemic. All these patients have high lipids, and there's a known antagonism between lipid uptake and glucose uptake through various mechanisms. Yeah.
This is what's causing the pH effect you're seeing.
Not having glucose opening the spigot for lactate. It could be.
Like starving.
Yeah, it could be. It could be.
Secondary to the-
Yeah, 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 it could be. It's formally possible. And one of the ways we will answer this is we are now we're currently repeating this study. We can't feed them, of course. We can't feed patients who are coming in for procedures, but we can infuse sugar, right? So we're repeating this study, infusing sugar and getting an insulin response. And that should tell us, actually, that should tell us.
Are putting patients not on statins?
Some are, most are. Yeah, most are. But most of the non-failing ones were also on statins. I mean, pretty much, you know, if you see a cardiologist these days, you're on a statin, right? Pretty much, yeah, right. So it's interesting, they're all hyperlipidemic. So there's this antagonistic relationship. So going back to these studies that were done in, yeah.
Do you have any interest in looking at VDL and LDL when you repeat this time? Maybe try to-
Very hard to measure.
Yeah.
That's the problem. Yeah, we're trying lipidomic studies right now. But the problem there is variability. It's very, very hard to measure with enough, you know, low variability and precision so that you can see these differences between A and V, because they're not big differences. Yeah, that's a great question. Yeah. This is an old study back from the, you know, we're back in the 90s again. So this is in, you know, again, 18-year-old volunteers. But what they did in this study, which I thought was kind of cool, is they infused increasing amounts of fat into these people as they were measuring A to coronary sinus. And as you get more and more fat, you can see that the glucose uptake goes down, right? So there's this kind of well-known inverse relationship, right? And I can tell you, this is 18-year-olds with increasing amount of infused fat, right? We're not infusing fat in our people. We don't need to infuse fat in our people. Almost all our patients were above this line, right? People are hyperlipidemic these days, right? So I actually think we live over here. So this could be the explanation why there's no glucose uptake, because we're looking at a different patient population. And what we learned 30 years ago is not applicable to today, right? So, you know, more on that. So we're doing the fed subjects, as I said, and of course, we're going to do HFpEF because you have to do HFpEF. All right, how am I doing for time? So the last interesting thing from this study was, oh, I'm okay. We'll go to 2, right? Yeah, or before 2. So the last interesting observation that I want to talk about was the amino acids. So what's going on in the tissue, inside that tissue with amino acids? And it turns out that all the amino acids are down, except for 3, leucine, valine, and isoleucine, which are branched chain amino acids, which have also gotten hot and interesting lately. And so that's what I want to talk about as the sort of third part. I want to talk more about branched amino acids. We've actually been interested in branched amino acids for a long time, for lots of reasons. But in the context of the heart, branched amino acids have gotten a lot of attention for a long time. So I'll show you in the next slide. But first, let me just give you the background, just to orient you to branched amino acids. I'm sure you all know this already, but just to reorient you. So there's 3 of them, right? Leucine, valine, isoleucine. They're hydrophobic amino acids. They're essential. You can't make them. You cannot synthesize them. You have to eat them. But despite the fact that you can't make them and you have to eat them, there's a lot of them in your body, a lot, right? So your protein mass is basically 20% these 3 amino acids. Okay, there's a ton of these things in there. And when they get catabolized, which they often do, it can be an important contributor to ATP, for example, into TCA cycle, right? And they all get catabolized. This is a little mitochondria back there. The entire catabolism happens inside the mitochondria. So this is a mitochondrial process. Catabolism is a mitochondrial process. The first step is this BCAT, which stands for transaminase. So basically, the amino group over here gets switched over to a hydroxyl group, so they become alpha keto acids. Okay, that's what these things are for. That's a rapid equilibrium reaction. There's no rate control there. This is the rate control step. The rate control step is this big complex, BCKDH, dehydrogenase. It takes a carbon dioxide away and it dehydrogenates. Okay, and at the same time, it puts a large coenzyme A moiety on the subsequent metabolite, so a large hydrophilic adduct, which means that once you get past this step here, all this stuff is stuck in the mitochondria. It can't get out. Just get this big, huge hydrophilic thing stuck to it. Okay, with one exception that I won't talk about today. And then it gets broken down further and it goes into the TCA cycle. All the carbons make their way into the TCA cycle. Okay, so the bottom line is it's a process that happens all in the mitochondria, and this is a rate-limiting step. That's really what you need to remember. Okay, and rate-limiting steps are regulated. Biology likes to do that, right? It regulates it with a kinase called BCKDK. So this is one thing I want you to remember, right? So the kinase is an exquisite inhibitor of this process. Okay, so more kinase, less flux, less kinase, more flux. Okay. So as I said, these things have gotten all the rage lately. This is, I always like to show this slide. This is a Google search frequency for branched amino acids. You can see in the orange line that there's been more and more interest in it over the years. It follows this fun sawtooth pattern that coincidentally, I don't know why, is precisely the same as a search for the word workouts, go figure. It's also interesting that this rapid rise happens on January 1st and coincides with New Year resolution, go figure. Okay, but that's not the reason why we're all excited about this, of course, but the epidemiology of branched amino acid is interesting. So it turns out that branched amino acids plasma levels are elevated in people with heart failure, and that's been shown in multiple different studies now, dozens and dozens, right? And we were not the first to notice that when you open up a failing heart and you take a tissue out, that the levels of branched amino acids are elevated, and that the expression of the genes in that catabolic pathway are down. Okay, I didn't show you that, but they're down. And so that has, and this is 2 studies that have shown that as well. So that's led this hypothesis, which is currently dominating the field, which is that there's a problem with catabolism of branched amino acids in a failing heart. And that's causing an elevation of branched amino acids in the failing heart, and that's causing problems. And that maybe this is a therapeutic target. Yeah.
How early is that?
We can't tell in humans, of course, right?
Yeah.
But in like in mice and rats, it's actually pretty early.
Okay.
Yeah, like if you tack, it's a pretty early event. Yeah. Yeah, in the mouse models and rat models, it's pretty early. Yep. So you know, it's led to this hypothesis that, you know, less catabolism in the heart of branched amino acids is bad, and more catabolism in the heart is good. Okay, and there's actually a lot of really good evidence for this. Sorry, I need a little water here. There's actually a lot of really good evidence for this. So, you know, more branched amino acids bad, meaning you can put animals on a high branched amino acid diet, or you can delete the phosphatase, which antagonizes the kinase. And then you can test this in multiple different models: transverse aortic banding, you know, ischemia reperfusion, myocardial infarction, all sorts of different models in rats and mice. You constantly get the same answer, right? More branched amino acids bad. And the converse is true as well. So if you remember that I told you the kinase inhibits the rate limiting step, right? There's a really nice inhibitor to the kinase. So if you inhibit the inhibitor, you increase the flux, right? Right? Less inhibitor, more flux. So this is a way to increase flux of branched amino acids. When you give this to animals, their branched amino acid levels in the plasma drop because they're increasing their flux. They're increasing their breakdown. And when you do that in the context of lots of failing models, they all improve. Myocardial infarction, ischemia reperfusion, pressure overload, multiple labs, literally 6, 7, 8 labs across the world. There's just no doubt that this is true. Okay, so people are feeling pretty confident about the hypothesis, but again, our friend Danielle was not. And so she's gonna test this hypothesis because nobody had really tested it. And this literally came out like 2 weeks ago. So here's the model, right? You have an insult, let's say a myocardial infarction. It decreases the expression of those catabolic genes. So you pile up the upstream stuff, the branched amino acids. You know, you have decreased flux, you pile up the upstream stuff, and that causes heart failure. And you can reverse the process by treating with a drug that improves catabolic flux in the heart. That's the idea, right? Okay, do we believe this? Well, I already showed you some of the tissue metabolomics and some of the RNA-seq that says, yes, these genes are down, check. And yes, the BCAAs are up in the heart, check. Okay, so we're good with that. Now, what about this question, right? Let's talk about the catabolic flux in the heart, BCA catabolic flux in the heart. Well, the first thing to remember, I showed you this already, is that the heart barely consumes any branched amino acids at all, right? It likes to consume fat and ketones and lactate. Here's the branched amino acids, a little tiny sliver over there. So it's already a little hard to imagine how that tiny sliver would matter. It's possible, right? But it's not a lot, all right? It's not a lot. And it's true in animals as well. So this is a type of study that we routinely do in the lab now, which is a tracer study to look at steady state, quantify steady state use of various different substrates by the heart or really any other tissue. So what we're doing is we have a mouse that has two catheters sticking out of it. One's in the jugular and one's in the carotid. And we can infuse things into the jugular and we can take things out from the carotid. And we can do that while the mouse is perfectly awake and happy and walking around. So it allows us to do very sophisticated studies, like, for example, infusing a little bit of 13C labeled, heavy isotope labeled, not radioactive, just heavy isotope, right, labeled branched amino acids. And you trickle that in there and you let the system go to steady state. And then you measure where it went, what tissues it went, and how much. And then you can do math from that. And you can actually quantify who's burning branched amino acids and how much. Okay, it's a very sophisticated modality. We use it a lot. I'm just going to show you one experiment, which is this one. So let's look in a heart that has undergone a heart attack. And let's see, does branched amino acid catabolism go down in a heart that has, right? So the way we do that is we say, well, let's follow these carbons into the TCA cycle, which is where they end up if catabolism went up, right? So here's our two examples, succinate and malate. I don't have this labeled, I apologize, but you know, blue is no MI and red is yes MI. First thing you'll notice is not much. I told you there's not much branched amino acids going in. And after MI, it goes up, not down. It actually goes up, right? Totally the opposite of the hypothesis that's out there, which tells you that gene expression isn't the be all and end all for flux. There's a lot of other things that dictate flux in metabolism than gene expression. Right, very, very important point. So this is not fitting the model, but still, you know, I did tell you that BT2 works in a gazillion different models, and we've done it ourselves. So BT2 definitely works. Inhibiting the inhibitor, you know, activating catabolism definitely works. You know, I showed you that, you know, schematically, and we've shown it too. So you know, what gives, right? This definitely works. But this is going the other way. So what Danielle wanted to do is she wanted to really nail down the question of, is BT2 working via targeting the heart, right? So it's inhibiting BCK-DK, but it's doing it all over the body, right? The hypothesis is that the way it's benefiting the system is by inhibiting in the heart, but we needed to prove that. And the way to prove that is to basically delete BCK-DK just in the heart. It's the same thing as giving BT2 just to the heart. That's the experiment, right? So that's what she did. We had to make brand new alleles because they didn't exist. So the CRISPR-Cas9, new floxed alleles. So here's a cardiac knockout of BCK-DK. So you could see BCK-DK is gone. And because the kinase is gone, then the phosphorylation site is no longer phosphorylated on BCK-DH. You can see that right there, right? So that spigot is now open, right? Because it's an inhibitory site. And we can prove that that spigot is open because we're doing our little infusion studies, right? And you can see that all the intermediates of the TCA cycle have much more incorporation now in these animals because the spigot is open. So we got more branched amino acids, right? So the prediction would be that this would be helpful in a heart failure model if the hypothesis is right. And the answer is does nothing, no difference whatsoever, right? It is of no benefit whatsoever, right? Experiment's been done a whole bunch of times. So that's not how this is working. That's not how BT2 is working. So now the story got more complicated. And I'm going to skip data and just show it in schematic form because it's more of the same type of data. So possibly it has nothing to do with flux and it has to do with just levels of BCAAs somehow. First, this made us worry. Always got to worry about this. This made us worry that maybe BT2 is not working through BCK-DK. Maybe BT2 is inhibiting some other thing off target, right? That was a concern. So we did the same experiment in animals that completely lacked BCK-DK. The whole animals got no BCK-DK. And if you do that, then there's no effect anymore. So BT2 is definitely working through BCK-DK. When you read about inhibitors and small molecules, you should always worry about off-target effects. There's no small molecule out there is perfect. We actually know BT2 has off-target effects, but they're not at play with this phenotype. But that's something you should always worry about. So maybe it has something to do with this, right? With low BCAAs. So we decided to lower BCAAs in the whole system by activating catabolism somewhere else than the heart. Okay, and we picked the muscle because I'm not showing you, but in a previous study we had shown that the muscle is really the main sink for catabolism. So we open the spigot in the muscle. Now the muscle is consuming a whole bunch of branched amino acid. Plasma levels of BCAAs go down, and then we do our MI studies, and the answer is nothing, no benefit whatsoever, despite the lowering of branched amino acids, just like PT2 does, right? So somehow this is working in a different way, not at all the way that we thought. So in the last few slides, I'll tell you where that's landed us. So it must be working over here in a different way, and we don't understand why. And we scratched our heads. We have to go back to the drawing board, right? We have to say, okay, we have all these things where it works, clearly works, and that's what it's doing, and it's working through BCK-DK. We know this part, right? So what do all these models have in common, right? And they're pretty different, right? They have pressure overload, myocardial infarction, ischemia. They're pretty different. One of the things they have in common is that there's a maladaptive remodeling after the insult. And that maladaptive remodeling involves lots of things, but one of the things that it involves is vascular reactivity and vascular remodeling, right? And we know this in part because, as I said at the very beginning, pretty much all the drugs we use are actually targeted against the vasculature, right? ACE inhibitors and ARBs and even ischemic reagents like NO donors are all targeted against the vasculature. So we thought, well, let's look at that. Maybe it's as silly as that, and it turns out that it is. So we looked at blood pressure. So we put little catheters inside the mice and measure their blood pressure in their aorta. And then we do it before or after giving BT2. And you can see they drop about 10 millimeters of mercury, right? Systolic blood pressure goes down, diastolic blood pressure, mean blood pressure goes down. And what's interesting is that as I'm sure you all remember, that blood pressure is regulated in part by nitric oxide being secreted from the endothelium to regulate smooth muscle tone. Now, that's not the only thing that regulates blood pressure, but that's one thing, right? And this is independent of that, because if we take NO out of the picture by giving an inhibitor to enos, then the pressures are higher because you got rid of the NO, but you can still lower the blood pressure by giving BT2. Same, right? So it's an NO independent, right? We don't understand. I'll tell you right now, we don't yet understand how it's working, but it's NO independent, which makes you think that it's probably a direct target on smooth muscle cells. And this isn't just, this was so surprising to us that we were like, okay, maybe there's something weird about using these caths in these animals. So we did formal telemetry experiments that I'm not showing you here, but you can implant telemetry, measure blood pressure in ambient mice walking around. We get exactly the same result, right? So that was pretty interesting. So is it autonomous to the vessels themselves? So to do that, we teamed up with Rick Asoyan, who helped us, take these we picked carotids, we could have done other vessels, and you take them out and you mount them on these little apparatus, you fill them with fluid, you bring them up to normal pressure, and then you challenge them with a constrictor, phenylephrine, right? PE, phenylephrine. So you say, okay, constrict. And you could see that the BT2 treated animals didn't constrict nearly as much. So they're somehow resistant to constriction, which explains why they have a lower blood pressure, right? On the other hand, if you give them acetylcholine, which is a vasodilator that uses NO as its signal, right? That worked fine. So the vasodilation is okay, which makes sense because NO is not in this picture. So it's a constrictive defect of some sort. That's as much as we know right now. We don't know more yet, right? So the last thing I'll show, what am I doing for time, is we always like to, whenever we see observations in mice, we came to the mice because we saw observations in humans. We like to go back to the humans to see if, okay, is this relevant to humans at all? So this was interesting. There's all this literature on branched amino acids out there. There's all this epi out there. How come nobody noticed before that branched amino acids might, correlate with blood pressure? Well, it turns out that people had. It's just that nobody's paying attention to them. So here's one study out of the Netherlands that looked prospectively. This is a different study, but they piggybacked on the study. They basically looked at metabolites at time zero in their study, and then they followed their patients longitudinally and tried to see if the levels of branched amino acids at time zero predicted who was going to end up getting hypertension down the line, right? And it turns out that it's quite predictive. So this says survival probability. That doesn't mean they died. That means that they survived without hypertension. So your chances of surviving without hypertension is much, much, much lower if you're in the top quartile of branched amino acid levels at time zero. Okay, it's kind of a prospective study. This is a case control study out of Japan. They basically compared hypertensive people to well-matched non-hypertensive people. And again, branched amino acids were the #1 signal that was elevated in the hypertensive. Now, this is all correlative, of course. It's hard to do causality in humans, but there is one trick that gets you close to causality in humans, which I'm sure you've heard of, which is Mendelian randomization studies. Okay, so we did that. We teamed up with Michael Levin, who's in the Penn Medicine Biobanks, really brilliant guy. And we did a Mendelian randomization. So what do we do? So we take GWASs that are out there that can tell us which SNPs in people correlate with elevations in branched amino acids, right? So small, single nucleotide polymorphisms that correlate with elevations in branched amino acids. And then we take that signature, that SNP signature, and we go to the UK Biobank where there's 1 million people with SNP information, and we say, do these SNPs that we know cause elevations in BCAAs, do these SNPs also alter blood pressure? And the answer is they did. Pretty much every marker, diastolic, MAP, mean systolic, et cetera, pretty much all of them were slightly elevated if you carried this SNP signature. Right? So that's pretty good, actually, in my mind. And in fact, if you look at these SNPs and you say, let's just focus on the SNPs that make sense. In other words, they're next to one of the enzymes that we know is involved in the catabolic pathways, then the signal gets even better, right? And the p-values get even better. So this is as good as you're going to get, I think, short of a randomized trial to show that that this is a real effect in people. So we always like to do that. So, I think this is all happening over there, and we're going to pursue this further. It also begs the question of, BCAA has also been involved in other things like insulin resistance. I actually kind of think that now maybe this is also the answer to insulin resistance. I can tell you that there's at least 4 different pharmaceutical companies that I know that are working on this and making drugs that are BT2-like to use in people. And they're doing that despite, I think, not understanding how this works, which is, not the first time that happens, of course. But I think, you're better off knowing what you're doing than not knowing what you're doing, if you can pull it off. So that's it. That's what I was going to say. So I talked about 3 different studies, 2 of them which just came out, a couple of them in humans, 1 in mouse. So the bottom line here is that branched amino acid oxidation modulates vascular reactivity and blood pressure in a way that we don't totally understand yet, but which might explain a lot of its effects on the heart and on resistance. And we don't understand it, but there's still clearly a therapeutic opportunity. So I'm not telling these pharma to stop. I'm telling these pharma to give me money so that I can study it more, so that I can tell them how it works. That's what I'm telling them, right? We don't really understand exactly how it works. And then, I like to make this point as much as I can. I love studying mice, but just have to keep in mind, it's not the same thing as people. This is all the people who did the work. It's a fantastic team. I love working with this team. Danielle is, is now in the words, and I'm going to do a physician scientist program in cardiology probably. And Emily is going to do a postdoc in Germany because she decided she was sick of the US. And I'll take questions. Thanks for listening.
The myocardium doesn't consume that much virtually nuances. How much does the vascular muscle?
We don't know.
You haven't checked it.
We don't know. It's actually hard to do because there's no real smooth muscle organ other than the uterus, but I'm not really sure that's going to be representative. And cell lines are cell lines. Cell lines are problematic. So we're going to do primary smooth muscle cells, right? And then we're going to try to do tracing in vivo into, let's say, the aorta, where at least half the cells are smooth muscle cells, right? So we're going down that road. Yeah.
So what causes degradation of protein in the fatty part? Is it related to mitosis or?
Yeah, I don't know, right? Yeah. What's degrading in the heart? Yeah, I don't know. We tried to do this. I don't think this really works, but did this just turn off? Oh, we tried to do this trick where we looked at the ratios of essential amino acids that were getting secreted from the heart, and try to figure out from that what kind of protein was generating it, because not all proteins have the same ratios of amino acid content. And it was interesting. This is just a complete conjecture, but it best matched myoglobin. Yeah. And so I'm wondering if myoglobin might not be, this is total crazy idea, but I wonder if myoglobin might not just be in part, a repository of amino acids. Yeah, yeah. I mean, why not? It's a repository of oxygen. Why not a repository of amino acids?
I guess you'll see too much infusions too, if it's like autophagy for fasting.
Right. So we have to go back to the. You mean in the humans?
Like, yeah.
Yeah, but it's hard to. I mean, we don't know what's going on inside the heart. Right. We don't know what's going on inside the heart. One of the things we're trying to do now is to look at also look at proteins that are getting secreted and coming out, right, in those studies. Proteomics is harder than metabolomics in plasma, it turns out, but and that might tell us something, actually. Yeah.
Since the branch amino acid is
That might not be on. Is that on? Yeah.
it's only oxidized in the mitochondria, could this be like, really reflecting the whole body mitochondria function under the condition of heart failure, that skeletal muscle, liver, and all these vital organs may have problem with the mitochondria, and branch amino acid level in the heart and in the system is just a reflection of that.
Yeah. So the question, if I can rephrase the question, is why are branched amino acids elevated, right? And I think that's a very likely proposition, that there's a whole body defect in BCAAs that may be related to a mitochondrial defect, but it doesn't explain the benefit. It doesn't explain the nefarious effect that it has on the heart, because we were able to drop the branched amino acids and it made no difference. But I think you're right. My guess is that's right. Yeah. Right. Somebody beat you to the mic.
Sorry, I'll be fast. I just wanted to return to the human heart and the fuel use in a healthy heart versus a failing heart.
Yeah.
If you could talk about what you might expect to see for cardiomyopathy driven by something other than ischemia, like virus-induced or something like that.
Yeah. It's a great question. So in the ACS studies, so the first study, it was a mixed bag of low ejection fraction. Some of them were ischemic, some of them were non-ischemic, but they were all compensated, right? They're all people on meds showing up, euvolemic because otherwise they weren't going to do an EP procedure on them. In the second study, we really focused on just non-ischemic dilated cardiomyopathy. When we looked at the tissue, for the sake of homogeneity, really, I didn't expect the p-values to be that good. So I was afraid that we weren't. So we're going back and doing it in the ischemics, and it may be different. And I think that would be fascinating if it was different. Because I'll tell you that when you look at, so we've done RNA-seq on both ischemics and non-ischemics, and I'll tell you they don't separate, which is interesting. So I think these, it's a caveat of these studies. These are end-stage, somewhat burnt-out hearts, or burnt-out hearts because they're getting a transplant. And so there's sort of I think there's a final common pathway where they kind of all end up. They obviously started differently. So it's a caveat. Maybe the metabolism we're looking at is it's not exactly the metabolism we want to be looking at. We need biopsies, but nobody biopsies anymore. Yes.
So first, just a quick comment. Your mouse does not equal human. I guess maybe also on there it could say that 18-year-old healthy human does not equal 60-year-old human. 100%. Yeah, but besides that, in terms of earlier on, you had mentioned the metabolism at night, and I'm wondering if that's a real circadian rhythmicity or related to sleep per se. Particularly in heart failure patients where there are sleep problems.
Yeah, where their circadian rhythms are off. Yeah. That's a great idea. I don't know how we would study that other than to do ACS at a number of different time points. And yeah. But Martin Young, who's at University of Alabama, I don't know if you knew him back. He's doing a lot of circadian studies in the heart in rodents. Right, in mice, and there's clearly very strong rhythmicity, and in particular with respect to branched amino acids, he's got this crazy story now, where if he feeds branched amino acids I'm going to get this wrong probably, but if he feeds animals branched amino acids in the evening, they get significant hypertrophy, but in the morning not, for example. So there are really interesting circadian links here. Yeah, 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 yeah. Yes.
Got a question for you. So going back to that earlier study with humans with heart failure and comparing them to humans with atrial fibrillation, we looked at the AFib. Sorry, we looked at the AFib patients as healthy controls. But given that they had atrial fibrillation, do you think there was anything confounding in that study?
Yeah. That's a great question. I mean, obviously, that's a caveat of this study. This is the pragmatic reality of doing human studies. You do what you can. We don't routinely bring completely normal people to the EP lab, right? So we wanted to be as homogeneous as we could. So everyone was there for AFib ablation, right? Both the normal EF people and the reduced EF people. So it's not a confounder in the comparison of the two, but it's certainly not a totally normal person, right? But we excluded anybody who had, hypertrophy on their echoes or had evidence of diastolic dysfunction or HFpEF on their echoes. So we tried to make it, other than the AFib, we tried to make it as normal as possible. But you're completely right. There's a reason they have AFib.
Would you ever consider, so I was doing studies a while back in a lab and we were using patent foraminal valley patients because they're getting catheterized as well.
Yeah.
So they're almost a perfectly healthy human to get sampled.
Yeah, it's a good idea. We should probably do that. The other example is AVNRT ablations, right? Same, basically completely normal. They just happen to have a little bypass that needs ablated, right? The problem was the numbers. We only, while we got 120 of these, we actually did get like 5 of them. But it would take years and years and years. Yeah.
Thank you.
Hi, Zolt.
Yes. Oh,
Sorry.
Oh, yeah.
Great talk, by the way.
Thanks.
Really enjoyed it. I just had a question in the very last part where you were talking about that the lowering of blood pressure wasn't NO related. Does it have to do with the endothelium? Do you have any idea? Have you done?
Yeah, we don't know yet.
You haven't done any.
Right? It could be
ex vivo?
endothelial dependent, just not NO. That's not the only way that the endothelium talks to the smooth muscle cells. There's pressors, endothelin, et cetera. And the simple way of answering that question is to repeat the vessel experiment, but strip the endothelium.
Right.
So we haven't done that yet.
Okay. Interesting.
Or it could, of course, be direct on the smooth muscle cells. Now, I favor direct on the smooth muscle cells because of the phenylephrine result, right? Because the phenylephrine talks to smooth muscle cells. At least that's the dogma, right? And there's a defect there, so it's downstream of endothelium. So I've got my money on that, but we need to do the experiments. There's another. Oh, here. Yeah.
Sorry.
Dr. Rogers, to you. Is there any work kind of expanding on this, like with potential methods like indirect calorimetry, looking at like full body energy partitioning and utilization, and like, is there any correlation with that to the heart and like metabolic flexibility holistically in that of the heart?
You mean in the context of heart failure?
Yeah.
That's that must exist, but I can't pull it off the top of my head. But people must have looked at that.
That's interesting.
Yeah, I'll go back and find that.
So
If that hasn't been done, then we'll do it because it's a really good question.
Yeah, it's not that hard either.
But it's such a good question that somebody must have done it.
Well, because the metabolic stuff you're doing, I'm like, that seems really hard, but indirect calorimetry is easy.
Yeah, no, it's true. Indirect telemetry is easy. Yeah. It's a good question. Yeah. Yes.
So regarding the ketone body utilization pathway, you said you weren't entirely sure where they were coming from, but it was thought to be the liver. Is that
Probably.
correct? Okay. I don't know if that's possible for you to do, but would you be able, I guess, the next study that you replicate to also catheterize like the artery and veins or major veins leading to the liver to measure ketone levels? Would that be something to
Yeah,
include
I have
this?
this is a great question. I have this conversation with the cath jockeys all the time, right? Where can I convince you to put the catheter? Right. The kidney is another interesting one. And that would be much easier. It's a much bigger vein and it's right there on the way up. But even that would add, it would add some risk and a fair amount of time to the procedure. So they're not excited about it. The liver is a problem because the liver's got 3 vessels, right? So it's complicated, right? It has got portal, vein, artery. And getting to the portal is, nearly impossible, right? I mean, there are some ways, but they're pretty invasive. So the liver, I don't know. It's not clear to me that we're ever gonna, The other place I really want to do is the brain. So we actually, believe it or not, we've done a few. We've done a few. I managed to get interventional radiologists to get excited about it. At first I thought, oh, it's going to be really easy. We'll just go to the ICU and drain, there's all these people with IJs, right? That's easy. The problem is that the IJ drains the brain, yes, but also the face. And the face is very vascular, right? So it doesn't work. So you have to thread a way up to the bulbus up there. So, but we've done like half a dozen of them. Yeah.
Thank you.
Yeah. Thanks for the question.
We've got one question from somebody online.
Oh, good.
Do you think follow-up studies to your research could include a dietary component? And if so, what would you want them framed like?
Oh, that's a good idea. That's a good, yeah, I mean, we could. That's going to be much more convoluted, but we could. I mean, we would like and are working on trying to do this, not the answer to the question, but on doing this with and without SGLT2 inhibitors on board, because that's all the rage. And we're actually probably going to do that. The diet is a great idea. I guess the first thing I'd want to see is somebody who doesn't have a BMI of 30 or doesn't have hyperlipidemia. So maybe if we could, get somebody to go on a diet for a while before we do it, I think it would be really interesting. That's where I'd start, I think. Yeah. It's a good idea.
All right. Well,
All
I
right.
think that's
Thank you all for all your questions. I appreciate it.
Thank you again.
Thank you. Yeah.